I'm really excited to share with you some research that we've been doing in our Refinement, Enrichment, and Advancements Laboratory here at the University of Michigan, and I'll be presenting some research results that were conducted by myself as well as Dr. Vanessa Oliver. Assessing post-op pain in mice is really critical for being able to provide optimal clinical care and welfare for the animals in our charge, as well as the translation of those results to our human patients. But it can be exceptionally challenging. They're prey species, and so they have a natural drive to suppress pain behaviors, especially in the presence of observers, and we have limited time to assess a large number of animals in our vivaria. So what should we assess for when we're trying to detect if pain is present in our rodent species? There's two general areas we can look for: presence of new pain behaviors, and these are good because they're pain specific, but they tend to be a little less obvious. We can also look for loss of normal behaviors. These are a lot more obvious and easier to observe, but have the potential to be less pain specific. As our first new pain behavior, let's look at grimace scales. Grimace scales first came on the scene in around 2010 with the mouse grimace scale and have since developed into the rat, rabbit, and some large animal species like horses and pigs. They've been found and validated to be able to detect postoperative pain and alleviations of pain with various analgesics. However, they can be challenging to use in rodents cageside. For example, mouse grimace scale has been shown to be significantly lowered when it's scored cageside versus a video score that's taken with the person outside of the room. So this was a publication that recently came out from University of Newcastle and PLOS ONE, where they looked at mean grimace scale scores as they were applied by a cageside observer versus taken from a photo in which the human wasn't in the room. And we see differences between different strains, C57, C3H, CD1, valve C, and also very differently between live scores versus photo scores. Another new pain behavior we could look for is ethograms, or changes in behavior. In our lab, in the refinement lab, we looked at new ways to assess guinea pig pain. So we looked at post-castration guinea pigs, either just after isoflurane anesthesia or after the isoflurane anesthesia plus the castration surgery, and we were able to see specific pain-influenced behavioral differences after surgery as compared to after anesthesia. However, when we tried this cageside versus video scoring when the observer was out of the room, we saw significant differences in our scoring. So as you can see from this graph, which was published over the summer in JLAS, you'll see that the mean frequency of the behaviors was significantly suppressed at all time points when it was scored cageside versus by remote video. So what about cageside measures of loss of normal behavior? Again, these tend to be a little bit more obvious. We refer to them as proxy indicators, or visual cues that indicate what activities the animals have been engaged in, or in this case, not engaged in, while not in the presence of an observer. One caveat is they have the potential to be influenced by a multitude of factors, including pain, but also potentially illness like fever, nausea, or even phenotype. So how do we conceptualize loss of normal behaviors? I think the easiest way to start is by thinking Maslow. So if we think about Maslow's hierarchy of need, animals might have eating, drinking, potentially grooming at the bottom of their hierarchy of needs, and if these are met, they can then move on to some higher orders like social interactions, sleeping with cohorts, playing, taking treats, maybe running on a wheel. And maybe at the top, we might think of this for humans, this would be engaging in art or music. For animals, it might be engaging in organization of their cage space, making a really great nest. And as animals are in pain, they start to lose these behaviors in the opposite order. So the very first behaviors that will disappear are things like nesting, or at the bottom of the pyramid where we see them not eating and grooming, this is when we start to recognize loss in body weight, dehydration, or an unkept hair coat appearance. So we really want to be able to identify pain at the top of that pyramid. Here's where a loss of consumptive behaviors come into play. So this was a graph from a Flecknell paper in 1999, where we see decreases in body weight, which come along with a decrease in food and water consumption if they've had surgery without analgesics. And again, we want to recognize pain at an earlier part of the pyramid. So how do we get up to that top tier? One idea is cage organizations. This is an ARIS paper from 2007, where they saw that nest complexity in cage organization after surgery was significantly reduced, but they could restore some of those behaviors by applying appropriate analgesia, in this case, Nsaids. So the mouse on the right has a very disorganized cage. The mouse on the left had appropriate analgesia and has this behavior restored. Next complexity is also a common measure that can be used for loss of a normal behavior. This paper was published in 2013, has a score of 0 to 5 from no use of nesting material, which is the score of 0 on the untouched nestlet, all the way up to 5, where they fully shredded the nestlet and made a nice cupped nest out of it. They found with this scoring system that 9 hours after surgery, less than 10% of the mice had manipulated their nesting compared to 75% of the mice at baseline. So this was something they could easily see a difference in that was obvious Cageside. And they found that with anesthesia only, the condition decreased nesting, but less so than surgery. So the data is here. And we can see their nest score is on the y-axis, and if they had surgery, their before score, which is the light gray, is quite high. It's around 2. After score, it was completely absent. They didn't have any nest to score. If they had anesthesia only, the effects of having anesthesia significantly reduced their nest score, but not nearly as much as if they had had the surgery. And you can see that if they have surgery with appropriate analgesia, it's about the same as if they never had surgery at all, which really is the goal. TINT score was a further evolution of this concept. 100% of healthy mice, 9 different strains, would retrieve a bit of nesting material and incorporate it back into their nest within 10 minutes. And they found that the likelihood of failing a TINT test was much higher after surgery. And this was published by Rock et al. In 2014. And in fact, there's a really great Joe video. So if you want to learn about TINT scores or how to score nest complexity, I'd highly recommend this article for a tutorial. Zone clearance test was the next step in evolution for nest consolidation behavior. They made this a more titratable score. It was published in the Journal of Pain in 2015, where there's 6 different possible areas that the mice can clear their nesting material from. And after 100 minutes, they've reliably cleared all of those zones and consolidated the nesting material into one corner. So to see a live score of what this would look like at baseline, there's 6 pieces of nesting material in the corner. And after 100 minutes, the mouse has successfully retrieved all those pieces and made a little nest out of it. After they underwent a painful stimuli, which in this case was acetic acid, they didn't consolidate any of those pieces. But if they treated them with an analgesic, in this case ketoprofen, they were able to mainly restore that behavior. So using proxy indicators, we tried to utilize all of these various types of methods in a post-op setting with mice, and we found it a little bit difficult for the purpose of testing analgesics, Analgesia Efficacy, because the nest quality was a bit subjective and we found it hard to standardize between different viewers. The zone clearance test was challenging because the mice would rarely decide to build a nest perfectly in the corner and sometimes would build in the middle of the cage, and then we weren't sure how to score how many zones had been cleared. And then we loved the TINT test, but it was binary, and we really needed something that was a little more titratable and had more variation in the scores so we could tease apart differences between different analgesics and their ability to alleviate pain in mice. So we decided to leverage some natural behaviors, and we looked at nesting behavior as something that could be objective, titratable, but we also needed a test where the nest could be built anywhere in the cage. We also thought about grooming behavior. We knew subjectively in evaluating mice after surgery that if they had eye lubrication, those that seemed to be doing poorly would still have really greasy eyes the next day and weren't grooming appropriately. And so we thought this is a really highly patterned behavior. It's reproducible. Perhaps we can leverage incomplete grooming from pain as a way to have a scoring system. So our first step was to look for products already available, one of which is Germ. So we chose the orange formulation, which is suspended in mineral oil. It's something normally used to teach kids how to wash hands. So kids apply it to their hands, wash their hands, and then show it under a black light. And if there's any Germ left, you'll get this glowing blue signal. We then applied a little bit of it to the top of the mouse's head, about 8 microliters, and watched how well they would groom it. So because we know they're always going to groom in the same pattern, it would reliably be distributed onto their bodies or transferred onto their bodies in a very specific way. So after putting it on their head, the very first place it's going to go is onto their paws. Next, we'd see it on their back paws. Then they're starting to get it onto their ears and eventually onto their nesting material as they manipulate nesting material in their environment. As they continue to groom, it'll start to come off in the opposite order. So we'll start to lose it in one of these locations, 5 through 2. Then only trace amounts will be found at the original application site where the concentration was highest. And finally, no Germ signal observed at all. This is an example what the live score looks like. So the mice in these pictures are anesthetized just because photographing the signal under black light is really challenging on moving animals. So these guys were under isoflurane anesthesia. So you can see where it was applied initially, where it was transferred to the toenails. It sort of looks like orange painted toenails on the forenails and hind nails. This is what it looks like as they try to groom it over onto their ears and onto the fringes of the nesting material as they hold it and pull it and make a full nest. Then the signal starts to go away. So here you can see they only have a little bit of glow germ signal on their toes and on the top of their heads. And then finally only a trace amount at their heads or the application site. And then finally when it's gone away completely. And we assessed this in baseline mice, a mix of C57 males and females. And we found that after an hour, they were already having a score of 4. And by 24 hours had reached the maximal score of completely grooming it off. We then partnered with a lab that was doing IBC ligation as part of a deep vein thrombosis model, and we assessed the grooming transfer after anesthesia, which was isoflurane, as well as after surgery. So in this graph, we see their highest grooming score achieved at various time points between 1 and 48 hours. The anesthesia line is in blue, so that was their score after isoflurane only, and the red was after the IBC ligation surgery. And we can see, at all time points, 1 through 48, there was a significant delay in grooming transfer after surgery as compared to after anesthesia. So we know that this was attributable to unalleviated pain. Next, we moved on to a Nest Consolidation Test, or NCT, where we sort of built upon the already published methods with the goal of being able to build, the mice being able to build the nest anywhere in the cage. So here we have a nestlet that we rip up into 4 pieces. We put 1 in each corner. And then we defined nest consolidation by moving those pieces to within a 2.5 by 3.5 inch area of one another with a score of 0 through 4 to consolidate. So here's 0 score when none of the pieces are near each other, a score of 1 when 2 of the pieces have moved close together, a score of 2 if 3 of the pieces have been consolidated, a score of 3 if all of the pieces have been consolidated, and then a score of 4 if they had a fully formed shredded nest. Again, we applied this to some animals that were just a mix of C57 males and females adults. And as before, after just 1 hour, they had already achieved a score of between 2 and 3, and by 24 hours had achieved the highest score. Then again, we evaluated the mice in the IVC study, both after anesthesia and after surgery, and we saw that the highest nest consolidation scores were achieved much higher or more sooner after anesthesia, so just after 8 hours, whereas after surgery, it took them about 35 hours. So here we do see a small effect from the isoflurane anesthesia, but the surgery really is the larger driver. So we know that unalleviated pain was the source of the difference in that signal. Okay, so just to pull this all together, these are our scores at baseline and after anesthesia only at 1 hour. So you can see if they had just baseline, they had a score of 5 or 2 for nesting. After anesthesia, slight delay in the glow germ, but no delay in nesting material. At the same time point after surgery, 1 hour after surgery, you can see the scores were significantly different. And so there was very obvious visual delay in both nest consolidation and grooming transfer. At 24 hours, the mice at baseline and after anesthesia had all achieved their maximal scores, but we still had significant delays in the animals after surgery, so scores of 4 and 2. So they were at where the animals were after just 1 hour after anesthesia, 24 hours after surgery. And then at 48 hours, we can see the nest consolidation behavior had totally caught up in the post-surgical animals, but we still had a delay in the animals that were doing grooming transfers, so still a score of 6. We then wanted to expand these tests to make sure that before we recommended them, they would work in the wide variety of situations that you find in your standard vivaria. So we wanted to know, will this work just as well in males as in females, in an inbred as well as an outbred? So we looked at C57s and CD1s, with various types of housing. So are they already have an existing nest? Are they socially housed? And we wanted to see the various effects of different analgesics on our test. So we had control animals who were just injected with saline, and then various analgesic treatments ranging from buprenorphine at 0.1 mg per kg sub-Q every 12 hours for 48 hours, including the preemptive dose. Carprofen, we actually put into the drinking water at 30 mg per kg. They got that drinking water bottle about 24 hours before surgery, and they continued that for an additional 48 hours after surgery. And then multimodal was the combination of the two. Study design, we studied the same animals under different conditions so we could control for inter-individual variability, so baseline, after anesthesia and analgesia, so that would allow us to quantify the effects of the anesthetic and analgesic drugs on our assessments. And then after a 7-day washout, to make sure there was no lasting effects of the analgesics or anesthetics, they would then undergo the surgery with the same anesthetics and analgesics. And in this case, the surgery was a simple laparotomy. The pain was then assessed at each of these time points, 2 through 48 hours after the baseline, anesthesia, analgesia, and surgery conditions. For assessments of postoperative pain, we wanted to anchor our novel assessments with some of the more traditional measures. So clinically, we often recognize unalleviated pain in the terms of weight loss, so they had body weight checks. We then did our grooming transfer and Nest Consolidation Tests, as well as more of the gold standard mechanical thresholds with electric von Frey. We also had locomotor activity, and so Neuroanalytical has a tablet system called Mousetrap, where you can put the mouse on a touchpad tablet, and it will count how many steps are taken, how fast they walk, things like that. And so we also used ambulatory parameters. Here we have our nesting consolidation test with scores 1 through 5. And we also needed to adjust the scoring system if they were going to have an existing nest. So the standard nesting material at the University of Michigan is an EnviroPack. So that's a tea bag-like material that holds about 6 grams of shredded paper. And so to make this scoring system work, instead of putting the nestlet pieces in the 4 corners, we put the nesting material on the opposite end of the nestlet and counted consolidation to be within 1 inch of the EnviroPack and then fully integrated into the nest. This is what the images look like for Grooming transfer in the C57s and in the CD1s. So you can see this had a score of 1 through 5, so we tried to simplify it a little bit for ease of Cageside use. So 1 is that there's a strong fluorescent signal at the site of application. 2 is that it's then gone to either the front or the rear nails, because it can be kind of hard to see the nails as they walk around the cage. So being able to see both the front or the back for that score helped make it a little easier to use. Then looking at the application of the Germ to the ears through grooming. And then finally having it slowly groomed off, so 4 losing signal at those sites, and then 5 having Germ no longer detectable. So for validation, this is our Nest consolidation score of the animals at baseline, after anesthesia and surgery at our various time points. The black line represents baseline, the gray line represents after anesthesia and analgesia, and the red is after surgery. So these are all of our analgesic groups collapsed. And here we see that there was a slight delay at the first time point, 2 hours between baseline and anesthesia, and that's indicated by the hash sign there. The stars indicate that there was a delay from surgery, and so we can see a delay in nest consolidation as a result of surgery for at least the first 12 hours. And again, this is for laparotomy. Next, we look at our grooming transfer scores. So we see our max grooming transfer score on the y-axis, and again, baseline in black, anesthesia, analgesia in gray, and red in surgery. Here we saw a much greater effect of the drugs on the behavior for grooming. So we saw some delays after anesthesia, analgesia as compared to baseline out through 24 hours. However, if you look at the black and gray lines, they're much closer together than you would say the black or the gray line from the red line. So the red line is surgery, and we had significant delays after surgery in grooming transfer all the way out through 48 hours. We found this test to be usable in both males and females. So here you see Nest consolidation score, female mice on the left, male mice on the right, and in both cases, they had delays in their Nest consolidation score after surgery in hours 2 through 8, and then it had normalized by 12. When we looked at grooming transfer scores, in both cases, we had delays in grooming transfer at the latter time points, about 12 hours through 48 hours. Interestingly, the female mice had a few more time points where they had significant delays as a result of surgery as compared to the males. And this kind of lines up with literature in terms of male and female differences with pain, acute pain. For pain effects, surgery versus anesthesia and analgesia for all of the measures that we took. With the females, we saw decreases in nest consolidation, grooming transfer, and at the one time point, 4 hours, we saw differences with mechanical thresholds, but we found no differences in females between anesthesia and surgery with body weight or locomotor activity. And this is important, especially if you're studying pain, that the nesting and grooming tests were actually more sensitive than some of these more standard measures that we rely on when we're assessing pain in these species. With the males, we saw a decrease in body weight at 48 hours and very similar differences with nest consolidation and grooming transfer, no differences with surgery with mechanical thresholds or locomotor activity. Moving on to our inbred and outbred, the CD1s had decreases in nest consolidation scores at our first 2 time points and C57s for the first 3. When it came to grooming transfer, again, we saw more of these differences on the later time points than we did the earlier. So 12 through 48 for the CD1s, and C57s had the additional time points of 2 and 8 hours where surgery suppressed their return to a normal grooming transfer. Taken together with all of the assessments that we looked at, the C57s had decreases in body weight at 24 and 48 hours, nesting consolidation 2 through 8, grooming transfer out through 48 hours, and 1 time point at which mechanical threshold was decreased. With our CD1s, we saw fewer differences, only 2 time points for nest consolidation, a few time points for grooming transfer, and no differences with surgery in terms of body weight, mechanical threshold, or locomotor activity. So again, more sensitive measures for nest consolidation and grooming transfer than some of our more typical measures. Moving on to different types of housing considerations. So singly housed versus pair housed for nesting. On the left, you see singly housed mice, and you can see a significant delay in return to normal nest consolidation behavior up through the first 12 hours after surgery. For pair-housed animals, it was really only the first time point. And this makes sense because if there's 2 animals working together to make a nest, they're able to collaborate and have that nest come together a lot faster than if a single animal is working on it by himself. So this let us know that nest consolidation test works best for singly housed animals and less so for socially housed animals. Happily, we were able to see with grooming transfer that it was equally usable whether the animals were singly or socially housed. We had been a little concerned with allogrooming, that they might groom off each other's glow germ and that they might reach a higher score faster, but that actually wasn't the case. And even when we used 2 different color glow germs to assess, we didn't see any transfer of one mouse's glow germ to another mouse as a result of allogrooming. So here we see singly housed mice had an additional time point at which their pain was suppressing grooming transfer as compared to the pair-housed mice, so potentially a social buffering effect or analgesic effect from being socially housed. Finally, we looked at nest consolidation, whether or not they had an existing nest, meaning that EnviroPack that was placed on the left-hand side. And we were able to see that whether or not they had an existing nest or were starting from scratch, the nest consolidation scores were reliably decreased or delayed after surgery for the first 12 hours. So we were really happy that the animals could both be socially housed and have nesting and be able to use a combination of these assessments. If you look at grooming transfer scores with and without a nest, again, we were able to reliably see decreases with our grooming transfer scores for about the first 48 hours, with the dropout around 4 to 8 hours, depending. All right, so taken together with all of our assessments, we see that pair-housed animals have a decrease in body weight, mechanical threshold, and locomotor activity, less so than we see changes with nest consolidation or grooming transfer. So here's the pair-housed, singly housed, and nest consolidation. So for our discussion, we were happy to see that nest consolidation was better at identifying pain at earlier time points, because this gives you a sense right after surgery how they're doing in those first 12 hours. Interestingly, grooming transfer was a little bit better at detecting pain at later time points as well as more time points. And this was really interesting. We're wondering if there's differing evolutionary drives to perform behaviors. Maybe it's more important in an acute stage to be warm, and so there's a drive to put more of your energies toward having a good nest than necessarily having a perfect hair coat. Maybe it's relative discomfort associated with the body posture required to perform the behavior. So in this case, we did a laparotomy. It might be very difficult to assume the upright posture and engage those abdominal muscles when you've had a laparotomy. Maybe you might see something a little bit different if the source of discomfort was, say, a craniotomy or a paw. So perhaps grooming transfer gets more to a dynamic pain status, which is it hurts when I do this, versus more of a static pain status, which is it hurts all the time, even when I'm sitting still. So nest consolidation and grooming transfer together might give us a really nice picture of both static and dynamic pain, acute early pain, and more longer lasting pain. Only minor differences were noted when we looked across all of our different sources of variation, whether it was sex, inbred, outbred. We did notice that grooming transfer can be used in socially housed mice, and that having an existing nest doesn't interfere with the ability to assess nest consolidation after surgery. When we look overall at our pain effects, again, we're seeing differences out to about 12 hours for nest consolidation and out to about 48 hours with grooming transfer. So let's look at our analgesic drugs. We have our saline-treated animals, which had decreases in their nest consolidation scores out through 12 hours. And when they were treated with buprenorphine, there was only a single difference there. Carprofen, a slightly later time point at 4 hours. Happily, with the multimodal treated animals who had both buprenorphine and carprofen, we saw no delays, and so we were able to completely control for pain-associated decreases in nest consolidation scores. When we look at grooming transfer scores, so saline treated is an A, we can see that there were many time points, 2 and then 12 through 48, where we saw decreases in grooming transfer with saline treated animals. We saw slightly fewer with buprenorphine, although still present at 12, 24, and 36, and carprofen about the same as with saline, and multimodal about the same as with buprenorphine. So still some room to improve analgesia efficacy according to the Grooming Transfer Test. So looking across all the broad spectra of different tests that we ran, with saline, we had decreased body weight at 24 and 48 hours. So as a clinical measure, it was able to recognize animals that underwent surgery but did not receive any analgesics. For nest consolidation scores, we saw decreases at 2, 4, 8, and 12. For grooming transfer, we had decreases in grooming behavior out through 48 hours. A single time point where unalleviated pain resulted in a decrease in mechanical threshold, and 2 time points at which locomotor activity was depressed. However, if we had only looked at body weight, mechanical threshold, and locomotor activity, we would have missed some really interesting changes. So for example, with carprofen, we're able to see that there are no decreases between those 3 assessments, and there is some improvement in nest consolidation because we only have a decrease at 1 time point. And grooming transfer, interestingly, the only difference we really saw was that the pain appeared to be alleviated at 48 hours versus 36. With buprenorphine, we saw even fewer differences between our surgery and anesthesia groups, showing us that pain was even more well controlled. And with multimodal, we had no differences in nest consolidation, so it was like they had never had the surgery. And then grooming transfer, we saw very few differences at those 3 time points. And again, that's more of a dynamic pain. It hurts when I do this. So I think it's good that we're controlling for that static pain, which is even if you're just resting, their pain levels were controlled. Overall, for analgesia efficacy, multimodal appeared to have the best control of post-laparotomy pain in mice, followed by buprenorphine. Carprofen, which again was about 30 mg per kg in the water, was much delayed and not nearly as good. And then saline was only slightly better or slightly worse than the carprofen. For conclusions, we were happy to find that nest consolidation and grooming transfer tests appear to be both valid measures for post-op pain. They were fast, easy to use, can be used Cageside, and they can be used in a diverse array of situations, whether mice were male, female, inbred, outbred, socially housed, singly housed, with or without an existing nest, and so hopefully can be applied to the wide array of situations that you find in your standard mouse vivaria. Multimodal analgesia appeared to have the most efficacious post-op pain, although there was some partial coverage noted with the grooming transfer test. So certainly it would be good to look into this more and see if we could play around with different doses or some different analgesics. Carprofen in the water, unfortunately, was not sufficient as a single-agent analgesic, and this sort of falls in line with other publications that have found things such as acetaminophen to be insufficient for post-laparotomy pain in mice. Future directions, we would love to be able to titrate some of these to figure out if there's a threshold for treatment. So if you see a score of X and it should be Y, that you should definitely pull the trigger and provide some additional analgesics. We'd like to be able to evaluate additional analgesic regimens and see if we can find more of a sweet spot where we're able to control for pain. We'd like to look at other surgery sites. As I mentioned, the grooming transfer test in particular, it might depend on where the site of the surgery or the pain is. So we did a laparotomy, which is pain from the abdomen. You might see something very different if the source of pain is the head or the paw, or a thoracotomy, meaning the chest wall. It would be really cool to see if this works with other rodents, such as rats, because they can also be really challenging to assess post-op. And it might have applications beyond just looking at post-surgical pain. It might be that we can use it to assess end-of-life care decisions and humane endpoints. So with that, I'd like to thank Dr. Vanessa Oliver again, who did a large amount of this work, and we could not have been sharing these results without her. Our Refinement and Enrichment Advancements Laboratory at the University of Michigan, with our lead technician, Sarah Thurston, observers who helped us assess these tests Cageside, Jenny Jones, Katie Simon, Dr. Kimberly Jen, Drs. Lucy Kennedy, and Drs. Tara Martin. And our funding came generously from the Grants for Laboratory Animal Science, or GLASS Awards, which are sponsored by ALAS, as well as Dr. Bob Disco and the Unit for Laboratory Animal Medicine. And with that, I'll take some questions.
Thank you, Dr. Lofgren, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. Now, let's get started. So our first question is, Dr. Lofgren, can you provide some additional details on how you chose the carprofen water dose?
Yes, thank you. That's an excellent question. We definitely were looking to provide some type of analgesic that wouldn't require injections, and I know delivering analgesics in the water is something that we often get asked about, and so this is something we decided to explore. We knew that from a publication in JLAS in 2013, they had already found that mice would readily drink carprofen in the water, though not so for meloxicam. There must be something about the taste. The mice weren't keen on it, but they would readily drink the carprofen. They actually tended to drink it more than the water. So we were pretty enthusiastic that that might be a good option. We then conducted a pilot study to evaluate the 10 mg per kg dose that was described in this JLAS paper. And what we found was here we have water consumption on the y-axis. We have time on the x-axis. Regular water consumption is in black, and in green is the carprofen water. So these are the animals that would undergo surgery and drink carprofen water. And we found that the animals were just as likely to be able to drink the carprofen water. In fact, they actually drank more of it as compared to regular water at several different time points. So at least that gave us full faith that they would consume it after surgery. And then we did PK analysis. So we collected blood, spun it down for plasma, and sent it out to some of our colleagues at the University of Colorado. And what they found was the plasma concentration remained high the whole time that the animals were on the carprofen water. So they received it 24 hours as their standard drinking water in a water bottle before they had any measures taken. And then time zero, then they had it for 24 hours, and we kept the water bottle on for 48 hours. And we also pulled a sample at 72 hours to see what would happen once you took the carprofen bottle away and just gave them standard water. So you can see that the levels were up around 30 micrograms per milliliter. And based on the literature that I think is derived from dogs, this was thought to be an adequate, efficacious plasma concentration for this drug. So we hypothesized that it was reaching therapeutic thresholds. But to test this, we ran it through a number of the same tests that I just went through, including mechanical threshold, nest consolidation, and grooming, and had some concerns. So I'll discuss those briefly. In that pilot test where we provided the 10 mg per kg of carprofen for nest consolidation scores, we found at baseline they had scores of about 4 between 2 and 24 hours. After surgery, those scores were significantly depressed for the first 12 hours, and so we had some concerns that analgesia wasn't being met. When we looked at grooming transfer scores here, you can see the black line baseline. So good grooming transfer scores and reaching maximal by 24 to 36 hours. But after surgery, we had significant delays at several time points. And so again, had some concerns that maybe analgesia wasn't where we wanted it to be. And then when we did mechanical thresholds with the Von Frey, we saw 2 time points at which there was a decreased threshold. So the animals were more sensitive to mechanical stimuli after surgery than they were after baseline. So even though they would readily drink the 10 mg per kg and we thought the therapeutic level had been reached and was steady state, it didn't show the analgesia efficacy that we were hoping for. And so we went into the literature again and looked at some other papers that had similarly been disappointed with carprofen at standard doses and chose a higher dose, 30 mg per kg, to assess. So these are the volumes of volumes of carprofen water that were consumed by the mice on our full study. And you can see here the black bar is the carprofen water. The multimodal are the animals who had carprofen water plus injections of buprenorphine. So they were on par and readily drank the water in both scenarios. And they had increases in water consumption as compared to baseline. So again, they were drinking it more than they would drink their standard water. We didn't see any differences in the amount of water consumed across any of our treatment groups or whether or not they had had just anesthesia or they had had a surgery. So they weren't drinking more, say, after having a painful condition and self-treating. We did have some concerns when we started looking at some of the blood parameters. So not so much with our CBC, but more with our chemistry. We had some increases in creatinine and AST, and I think that's because they were actually consuming potentially a higher dose than even the 30 mg per kg because they really liked the water. So I think caution is to be made when you're trying to use any sort of analgesic in the water, and that you need to make sure the animals are consuming it. That published PK values are helpful, but don't necessarily guarantee analgesia efficacy. So it's important not only to have the plasma levels, but also to do the analgesia testing to make sure that those plasma levels correlate to reduced pain. And then with Nsaids, as it turned out with carprofen, if the mice really like drinking carprofen water, perhaps it tastes sweet, they're going to drink it and potentially get a higher dose than you intend them to, and there might be some possible sequelae for different organ functions. And even at 30 mg per kg, we didn't necessarily see the efficacy that we wanted to see. So I think at this time, I wouldn't recommend carprofen water as at least a sole analgesic. But for future directions, it would be great to look at a lower dose and see if we could pair it with something like an opiate to have good analgesia efficacy without the risk of potentially having some off-target effects. Thanks.